Welcome back. So today we're going to start Unit 2, and you can find the resources for this linked below. And if you're using my webpage, then you would go to Interactive Lessons, AP Biology, and you can search by unit. So it's the cell structure and function. And what's interesting and exciting for me is I've actually added some accessibility colors. So you can have a play of that. Or you can have a dark mode as well and switch it around. So a friend of mine pointed out that she didn't particularly enjoy the palette on one of them. It offended her eyes, so I switched it up. And so we'll get going here with cell structure and function. Now, this is the where cells are introduced in AP. Now, a few things to think about in the beginning is the basic components that you find inside a prokaryote and a eukaryote. Now, once you've done that, we'll think about a comparison between the two. But we're probably all a little bit familiar with the things that we find, such as plasma membranes. In this one here, we don't have a cell wall, but you will find a cell wall in a plant cell, for example. You find cell walls in prokaryotes. They're made up of something called peptidoglycan, whereas in a plant cell, it would be cellulose. They both have plasma membranes. They're both going to have ribosomes, which are these little circular guys here. This isn't the best picture. It's a nice representation, but if you're looking for something a bit more detailed, then here we've got our ribosomes, but they're also depicted here as solid circles. But it's not too much of a problem. Sometimes I think when a diagram is maybe a little bit poorly constructed, it's an opportunity for us to think a bit deeper and show our understanding. So ribosomes are 70S in a prokaryote, and they'd be 80S in a eukaryote. I've got a nice comparison down here. For the Dora, the Explorer fans, here's a mnemonic device to help us remember exactly what we're comparing. So if you think of Dora, it gives us a basis. So DNA in a prokaryote is not found in a membrane, so it's not part of a nucleus, it's not membrane bound. In a eukaryote, it will be, and it's associated with proteins, so wound around a histone. It's linear versus circular in a prokaryote. We can see that here. Here's our circular nucleoid region. It's not membrane bound, whereas we have a double membrane. When we think about the origins of cells, which is a later unit that we cover, then we'll think about why these double membranes exist. And the idea is that prokaryotes came from eukaryotes, very briefly. In Unit 6, we think about introns. So an intron is a non-coding region of DNA. So what we mean by non-coding is this isn't that it does nothing. Sometimes it's referred to as junk DNA, but it's that it doesn't code directly for a protein. Now, there are no organelles in a prokaryote. By definition, an organelle is something that's membrane bound. The advantage of that is you can operate with a unique pH, for instance. So the little title that I gave this unit was Membranes by a Cell, the Right to Run Incompatible Chemistry at Once. So that means that you can have two different sacs. Like when I say sac, I mean bound by a membrane that have different pHs, different compositions. So pH is something that would define the activity of an enzyme, and some enzymes prefer or they all have their own very specific pHs. So you can operate little experiments at different conditions within that, and it makes for a more efficient cell that's able to carry out more complex processes. Now, a mitochondria is an example of an organelle, the rough endoplasmic reticulum. We've also got down here, you can see a Golgi body. Now this, I don't like the flow of this. I had to give up constructing it with Claude at one point. I'll come back to update it later. But it's like when we think about the parts of the cell in a eukaryote, what's really nice to do is look at the endomembrane system. And what we mean by that is these internal membranes that all have a part to play in the production of a protein. So if you think, we start with the nucleus, and the nucleus has DNA. The DNA will code for a protein. So a little mRNA transcript will move from the nucleus to a ribosome. Now, if it's going to be destined to be exported or used as inside a lysosome inside the cell, then that ribosome would attach itself to the rough endoplasmic reticulum. It's rough because it's got ribosome studded. Once it's been transcribed inside there, it will then and translated, inside there, it will then move via a vesicle. So here's a little vesicle, move to the Golgi body. The Golgi body, Golgi apparatus, the Golgi complex, it's got a few names. Its role is to then repackage it. And what we mean by that is add a carbohydrate, for instance. And it does a little bit of modification, which is not so important at this level. So we just think about it as adding a carbohydrate. Those carbohydrates, if embedded in a protein that gets used in the membrane, when we talk about membrane proteins later, they could be used as a receptor, for instance, or a way of indicating what the functionality of this cell. Now, it's really important to just know what an organelle's name is and its function. It's not something that needs to be lectured to you. It's something that you should memorize. And then putting it together is when you think about the endomembrane system. And if one part of it's missing, what is the end result? Is it that we don't get the end product, that we don't get a vesicle that's able to transport this protein anymore? I've seen questions where you're asking just to show the flow. So it's understanding how these parts fit together, that we go from the nucleus to the rough endoplasmic reticulum, to the Golgi, to a vesicle to be exported, for instance. Now, with this complexity means that we also have an increase in size. But having an increase in size does not come without its challenges. So a prokaryote is about 1 to 5 micrometers. And a micrometer is 1,000 micrometers to 1 millimeter, whereas a eukaryote is 10 to 100 micrometers. Now, make sure you go and learn all of those organelles and their functions. Now, you can find all of those in Cramly as flashcards. Please make use of that free resource. I think it's a really valuable one, of course, because I built it. And then when cells start getting bigger, the limitation is that the transport mechanism within is due to diffusion. So if we've got a cube that's 1 centimeter, and we're going to think about something called surface area to volume ratio. Now, the bigger the surface area to volume ratio, the faster rates of exchange are. So the surface of a cell is going to be directly proportional to the rate of exchange with components within and out. So like oxygen moving in and out, or carbon dioxide moving in and out. And the volume is directly proportional to the metabolism that's happening inside it. So the building stuff up, breaking stuff down. Respiration produces heat as a byproduct. If it's too big, then unfortunately that heat can't escape. I used to think about like those in class, we spoke about like bodybuilders and the amount the bodybuilder needs to eat to maintain their size, and that's because their volume has increased. And also you'll find that sometimes the very large bodybuilders are often very hot and operate at a very high temperature because the rate of respiration is very high within their cells. And because they have a lower surface area compared to their volume, the rate of exchange or heat loss is much lower. So as we increase the size, the rate of diffusion is much slower. Now there's a lab you can do where you put little jelly cubes that are stained with like phenolphthalein and acid, and you'll see them moving in, and you might time how long it takes for that block to change color completely. And you'll notice the bigger the block, the longer it takes, because diffusion is slow. And that means if something gets too big, then in this region here, like you could consider that anoxic, like not containing oxygen at the moment. If like the amount of time needs to get to the center, then you're going to have like buildup of toxic components, the waste products of metabolism, such as carbon dioxide, might build up to levels that are going to start affecting the pH, because carbon dioxide in solution becomes carbonic acid, for instance. Now, a nice way to demonstrate this is with a graph. There's a little one here just showing you as the side length increases, our surface area to volume ratio decreases, which you kind of think maybe that doesn't quite make sense. But I'll show you here with a sliding image. Now, if I increase that side length, you can see it gets bigger. And if we do a calculation, if we take that surface area and you chuck it in the calculator and you divide it by the volume, then you'll be starting to get this ratio here. If I do this divided by this, this is the ratio I end up with. And it gets smaller and smaller. And the big surface area to volume ratio is what we really want. for maximizing the rate of exchange. Now, some cells, in order to maximize this, an adaptation might be that they are flat, like pneumocyte inside the lungs, biconcave, like a red blood cell, or have projections like microvilli in order to increase our surface area there. And this is just showing you that with a small cell, the fusion can easily reach the center, but with a large cell in that same time frame, there's a huge proportion that's just left out. And then that's just not, it's counterproductive. Like oxygen's not going to get to any mitochondria there. Anything moving within a cell is there, like these small components that are going to be used in like respiration, they're moving by diffusion. And diffusion is a slow, random process. So surface area to volume ratio is really important. Within the AP, you might be asked to calculate and make comparisons between different models of cells that might be cylinders or cubes, and the formula will be available to you in the formula booklet. And it's just a simple, like you might need to work out like the surface area of the circle and then calculate what it is as a cylinder, for instance. But it's simply, when you're doing surface area to volume ratio, you do surface area, like here, 6 divided by 2.3 gives us our surface area to volume ratio there. And then you can make a direct comparison. So if we think, how does this all come together? And then I've mapped it out here for you. So when we think about the endomembrane system, we're thinking about the compartments, the components of a cell, like the rough endoplasmic reticulum, where the ribosome studied on it will produce the proteins that then move to the Golgi apparatus. And those compartments will also allow for a small surface area to volume ratio, which means that the reactions will happen faster within them. And a same trick, but in a smaller scale, is also our cristea and microvilli. So this is the smaller scale of the compartments, but the same trick is increasing surface area to volume ratio, which means that we can have a faster rate of reaction because everything's just brought closer together. So those chance events of them reacting is just increases. And then to finish off, for you to do at your own leisure, you have fill in the blanks and a few questions. And I'll link in a practice test question as well, a practice test. So you might have seen before on my site, I have under the multiple choice quizzes, like we're looking at AP here, cell structure and function. So you can find a 10-mark question here, 10-mark test here, and you can also find some different questions here. So you've got 16 questions there to help you practice. And just to look at the first couple. So a cube-shaped cell model of side 2 centimeters is compared with one of 4 centimeters. Which statement about the larger model is correct? Let's give you a second to think about it. Now, if you remember, it's an inversely proportional relationship. So its surface area to volume ratio is half as large. As that cell increases in size, and the volumes increase, is actually proportionally decreasing its surface area to volume ratio. So lysosomal enzymes work best near a pH of 5, while the cytosol, which is the liquid component inside our cell, cytoplasm is like all of the little components within it as well, but the cytosol on its own is just that liquid. This difference is possible because the lysosome membrane maintains a separate compartment. They're not all going to be B, just they happen to be there. So here's our sequence. Maybe you have to label this on the diagram. We go, remember, rough ER, vesicle to the Golgi, and it's missing out a vesicle here to the plasma membrane. But that's a common thing to do in AP. They're trying to trick you out. They're giving you part of the answer, leaving out a little bit of it, but it's not incorrect. It's the best flow out of the 4, which is a commonality in these AP questions. We might as well keep going. So which structure is found in both prokaryotes and eukaryotes? So you can do your DORA to think about what we've got there, like as a comparison. And we have ribosomes in both. So there is 70S ribosomes in prokaryotes and 80S ribosomes in eukaryotes. Cells lining the small intestine are covered with microvilli. The most direct advantage is that microvilli increase the surface area available for absorption. Correct. They're also, yeah, they will reduce the distance. No, they're just helping with the uptake. There is a smaller distance to the center of that microvilli, um, but that doesn't, like, where is that actually traveling to? It's not just going in there; it has somewhere to go afterwards. A student measures how far a colored dye penetrates identical agar cubes. Maybe you'll do this with your teacher, and the dye reaches the same depth in every cube. What this best supports the conclusion that, so give you a second to think, the proportion of volume reached falls as size rises. Boom. A constant depth means the shell reached is the same thickness in every cube. So it makes a smaller fraction of the larger cube. This is a classic lab that maybe you'll be doing with your teachers. So I hope you found today's lesson useful. Please feel free to check out also this multiple choice test here to quiz yourself. You'll see that you get feedback with each question, and feel free to check out all my other resources like Cramly. Okay, so ciao for now, and thanks for